Hello, hi, very happy good evening. So this is Krishnaveni and we have been talking about the story of life on earth, and that's biology. Yes, welcome to my lecture. So today we were talking about an important chapter, that is your molecular basis of inheritance. So this chapter has a lot of weightage. So among the genetics and evolution units, so this chapter bags a very important position. Yes, Prince, hello, very happy good evening. I'm good, thank you. How are you? Yes, Prince, I hope you had a good weekend, and thank you so much for joining my session today. So please do like, share and subscribe. So did you enjoy the rapid fire Menti from Mendelian Genetics, guys? Yes? So did you all enjoy? Okay, well, so we are starting a new chapter, so it's a new start. So this week we are starting a lot of new chapters. So today we have started molecular basis of inheritance. So tomorrow at 4 p.m. we'll be starting your human physiology. So this week it's going to be a very heavy week because you're going to start a lot of chapters. We're going to talk about some important chapters, important topics. So please pay attention. Okay, Prince, fine. So hi Anthony, yes, a very happy good evening. So thank you for joining my session. I'm good, thank you. How are you? So come on guys, please do like, share and subscribe. Okay, so moving to this chapter, molecular basis of inheritance. So what are we going to talk about in this chapter? So before we go forward and talk about molecular basis of inheritance, so I have an announcement to make. So today we are discussing important questions from the chapter cell cycle and cell division. So I have just completed this chapter, but I have not even discussed one question. So today we will discuss around about 30 questions or 25 questions from this chapter. So please do join me at 6 p.m. for this question session. So this is at 6 p.m. So guys, please do share the session with your friend. So ask them to join me as well. So Purnima, hi. Yes, a very happy good evening. So I guess you're new to my session. So thank you Purnima for joining my lecture today. So please do like, share and subscribe. Hi Keshav. Yes, a very happy good evening. So thank you Keshav for joining my lecture today. So how are you? I'm good, thank you. So come on quickly, please do like, share and subscribe. Okay, so this is lecture 1 of molecular basis of inheritance. So what are the topics that we are going to cover in this chapter? So here let me give you a short recap of the topics to be covered. So I have the topics of only today's lecture. So let's see if we can cover only them. Okay, so Abhinandra Nayaka, yes, hello, hi. So I guess you're also new to my lecture. So thank you for joining my lecture. I hope not to disappoint you. So please do like, share and subscribe. So today I'm starting a new chapter, that is your molecular basis of inheritance. So yes, and welcome. Hi Dharni, yes, I'm good, thank you. How are you? Yes, a very happy good evening, and thank you so much for joining my lecture. So please do like, share and subscribe. So what are the topics that we're going to look forward today in class? So we are going to talk about the most important molecule, the most important bug, you can say, that is the DNA. Yes, so how this small piece can go on for life after life, and how it ensures sustainability, how it ensures mutation, how it thus ensures variation and evolution. So this is overall a splendid molecule. So today we are going to discuss about this splendid molecule, that is the DNA. Okay, so then search for the genetic material. Well, so yes, I told you DNA is a splendid molecule, there is no doubt about it. But how did we come to know that DNA is the splendid molecule? Or how did we find out that DNA is the genetic material, and why not RNA or proteins? So search for the genetic material. So how did we go on, right? So then RNA world. So actually RNA plays a very important role compared to DNA, because RNA was much ancient. So we'll talk about why RNA backs an important position. So for example, though now you have new ministers, new people in the scenario in the parliament right now, so always Gandhiji is revered, right? So we have the photo of Mahatma Gandhiji in the currency notes. Yes, so your RNA is like Mahatma Gandhi, which played a very important role. But for the current system, you have ministers like DNA, which are important. Okay, so RNA is very ancient, but due to some reasons it could not sustain as a genetic material. So we will see. Okay, yes, Shimpi, hello, very happy good evening. I'm good, thank you. How are you? So thank you Shimpi for joining my lecture today. So please do like, share and subscribe. Ma'am, I have sent you a message in Telegram, so please see and clarify and find out. Yes, very sure. So I guess you were asking me about the neuronal potential, if I'm right. Yes, Yes, I will send you a voice message so that you understand the entire part of what happens. Yes, I saw it, Darni. Yes, I will do it. Ma'am, according to my calculation, my mark 564 out of 600 in public, but no use of this mark, only NEET is needed. Okay, all right, Keshav, so don't worry. So you will definitely do well with NEET. So hi Naveen, yes, after a very long time you're here. I hope you're doing well. So thank you Naveen for joining my lecture today. So please do like, share and subscribe. And I also want to tell you something. So you guys are going to have I have a surprise for you guys. So you guys will have some interesting short videos from me. Okay, so you're gonna have videos about snippets, about the chapter, about small concepts, and yes, do wait and watch for something very interesting as well from me. I have planned out something, so let's see if it works out that way. All right, so Keshav, when are you gonna give us treat? So you have passed your board exam, that is your public exam, with wonderful colors. So when are you giving us treat? So you are supposed to send me a cake, right? So when are you going to do it? All right, so we are going to talk about the RNA world. Then the most important process, replication. So from the chapter cell cycle and cell division, we know the DNA replicates. DNA replicates in the S phase, that is the synthetic phase, right? So how it's going to happen? So whether the cell has to invest energy, whether the cell has to spend all its energy, all its stamina. So how and why does this process of replication happen? So this we are going to see. So these are the first few topics that we will cover. So today let's try to cover from till at least the search for genetic material. It is not easy. You have quite few points to remember. Okay, so here we move forward to this chapter. So genetics, yes, as I have already defined genetics, as I have already introduced genetics. So here we go. So if I say the term genetics and if I say the term hereditary, so what is the difference between both? So hereditary is the transmission of characters, resemblance as well as differences from parental generation to the offspring. So now why do you resemble your parents? Okay, so for example, if I have 10 Labradors, right? So if a mother Labrador gives birth to a puppy, the puppy looks like any other Labrador, right? So they all look like dogs to us. They do not have much variation. But you and I do not look alike, but we resemble our parents. Why? What is getting passed down generation after generation? So that is your transmission of character. So that is what is known as hereditary. So it is resemblance as well as differences from the parental generation to the offspring. I am not exactly identical to my mother. Yes, I have resemblance as well as I have differences from my mother. So that is why my mom and me are not close, right? So this term is known as your hereditary. Okay, yes, so please don't send me food icons, then yes, it will stimulate all of our hypothalamus so that we feel hungry. Okay, fine. So then, how is genetics different from hereditary? So genetics is the study of hereditary, variation and environmental factors responsible for these. Okay, so hereditary and genetics are two different things. So hereditary is the resemblance of characters. So now if I have straight hair, I can say it is hereditary. My mother had, my grandmother had, so I am having this hair. So study of this hereditary is known as my genetics. So please do get these synonymical terms clear so that you do not confuse with some basics. All right, yes. So moving further, so the word genetics was coined by William Bateson. So he was the scientist who coined the term. So he is Mr. William Bateson. Yes, so you have one pair of chromosomes from your mother and one pair from your father, right? Alright, so now talking about the most important molecule, that is your DNA. So DNA is our genetic material. So it is a long polymer of deoxyribonucleotides. So in biomolecules, when we were talking about proteins, I told you proteins are polymers of amino acids, right? So similarly, DNA is a long polymer of deoxyribonucleotide. So please get it clear. So DNA is a type of nucleic acid. Okay, so nucleic acids are polymers of nucleotides. Okay, so DNA is a nucleotide and it is a polymer of deoxynucleotides. Done? Yes. Yes, that is also true. You can resemble your aunt, your cousin. Yes, everything is possible in genetics. Okay. Yes, so they are long polymer of deoxyribonucleotides. The length of the DNA is defined by the number of nucleotides that it has. Okay. So all of us know our DNA is like this. So we have a double helical DNA, right? So our double helical DNA, so I'll tell you what are these numbers. Right? So how do I define the length of the DNA? Suppose if I have a protein, so yes, the protein, how do I say the protein is this long? So I measure the protein in terms of amino acids, right? So similarly, for a nucleic acid, that is DNA, I measure it with the help of nucleotides, that is base pairs, that is pair of nucleotides. That is how I measure your DNA. Then this length of your DNA is characteristic of a particular organism. For example, humans have a particular pair of nucleotide or base pairs. Okay, fine. So now these are examples. So these are given in your NCERT in terms of lines and paragraphs. So I have tabulated it, so it's nothing new. Okay, so here we go. First, it's bacteriophage. So bacteriophage is a is a virus that infects bacteria, right? So your bacteriophage has a base pair of, if you look, it does, so here this talks about the number of base pairs, right? So this talks about the number of base pairs. So bacteriophage, a virus that infects a bacteria. So this has 5,386 nucleotides only. A bacteriophage, another one, this is bacteriophage 5 into 174. So this you call it as lambda. So this is 48,502 base pairs. Okay, so E. coli 4.6 into 10 to the power 6 base pairs. Okay, so this is important. So I am telling you, as the complexity moves higher, the base pairs, the nucleotides also increases. Okay, so now haploid content of human DNA, we are diploid beings, right? So we are 2n actually, but here they are only talking about the haploid content. Haploid content is 3.3 into 10 to the power 9 base pairs. So the diploid content is 6.6 into 10 to the power 9 base pairs. Okay, yes. So this is very, very important. So please remember, they might ask you in find the incorrect statement, find the correct statement, etc. Okay, yes, Antonia, I hope you find it very helpful. All right. Hi, Munna. Yes, hello, how are you? I'm good, thank you. So thank you, Munna, for joining my lecture. So please do like, share and subscribe. So this is about the first slide. Okay, so I hope you got it clear. So what is important in a DNA, or how do you measure DNA in terms of nucleotides? And each organism has a characteristic length of nucleotides. So if they ask you how many nucleotides a human being has, the diploid content of a human being has 6.6 into 10 to the power 9 base pairs. Okay, yes. Yes, Shubham sir, hello. So thank you for joining our session today. So please do like, share and subscribe. Okay. So moving forward, so today we're going to talk about the structure of a polynucleotide chain. Yes, Prince has understood. Okay, yes, Swami, a very, very happy good evening. So thank you for watching my session today, watching my lecture. So please do like, share and subscribe. So structure of a polynucleotide chain. So this we have already discussed in your biomolecules. So you know, so your DNA is a nucleic acid. So it is made up of nucleotides, right? So what are the components of your nucleotide? So we have a nitrogenous base. So we have a sugar molecule. And we have a phosphate moiety, right? So these are the things that are present. So nitrogenous base is of two types, that is your purines and your pyrimidines. So nitrogenous base plus your sugar is known as your nucleoside. So nucleoside into a phosphate is your nucleotide. Right, so this is the combination that I have discussed before. So your purines include your adenine and guanine. So pyrimidines include cytosine and thymine in case of DNA. Okay, yes, fine. So that's fine. So you are getting thorough. It's okay if you have learned it in chemistry as well. Okay, so in case of DNA, we have 4 bases, right? So suppose if my nitrogenous base is adenine. if my nitrogenous base is adenine, so in case of DNA, my sugar molecule is deoxyribose sugar. So first my adenine binds with deoxyribose sugar and it forms adenosine. So this is a nucleoside, right? Okay. Yes, Guha Karthikeyan. Yes, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. So this is important. So a nitrogenous base and a sugar are bonded by glycosidic bond. Right? So this we already know, it is bonded by N-glycosidic bonds. So now if I take another nitrogenous base, that is guanine, and if it's binding with deoxyribose sugar, then it becomes guanosine. Suppose if this binds with a phosphate moiety, okay, so it becomes adelenic acid. So since it binds with deoxyribose sugar, it is deoxyadenosine and it is deoxyadelenic acid. Okay, so this is how it happens. So here I have tabulated it. Okay, so this basic things I have told you. So nitrogenous base is linked to the pentose sugar, where to the OH of the first carbon of the sugar through N-glycosidic bonds. So this I have told you. Then the phosphate group is linked to the nucleoside via the 5' carbon of the nucleoside. So I have my nitrogenous base, I have my sugar. Right? So to the first carbon of the sugar, I have my glycosidic bond. Now to this my phosphate is binding. So my phosphate binds to the 5 carbon of the nucleoside and this binds to the phosphoester linkage. Alright. So here it is. Yes. Okay. Ma'am, aap Didi ko. Oh, so Shimpi is very slim. Shimpi is very slim. Oh, so Shimpi, see how caring Munna is. Okay, Munna, I will explain to Shimpi that eating is important. So you are a very good girl, right? But Shimpi isn't. Is that what? Alright, fine. Okay. So please remember this table. This is also equally important. So adenine binds with the nucleoside. So this is adenosine. So when it binds with the ribose sugar, so ribose sugar is in case of RNA and in case of DNA, it is deoxyribose sugar. So if it binds with deoxyribose sugar, it is deoxyadenosine. Okay, so now when it binds to a phosphate group, it is adelenic acid or it is deoxyadelenic acid. So this is in case of RNA and this is in case of DNA. Okay, yes. What is synaptonemal complex? Okay, so just give me 2 seconds. I will explain this table and then tell you what is synaptonemal complex. Okay, so then guanine. So guanine, when it binds with the ribose sugar, it is guanosine. When it binds with deoxyribose sugar, it is deoxyguanosine and it is guanilic acid and it is guanilic acid. Okay, so cytidine, this is cytosine. Okay, so this is cytosine. So cytosine becomes cytidine when it binds with ribose sugar and when it binds with deoxyribose sugar, it is deoxycytidine. It becomes cytidilic acid or deoxycytidilic acid. Finally, uracil or thymine, if it's uracil, it binds with ribose sugar, that is uridine. Thymine binds with deoxyribose sugar, that is deoxythymidine, and this becomes uridylic acid and this becomes deoxythymidic acid. Ma'am, why is this table very important? So they will give you a list of 10 compounds and they'll ask you to find nucleosides separately, nucleotides separately. So you should know how a nucleoside is formed and how a nucleotide is formed. So thus this tabular column is important. So please pay attention. Okay, yes. Okay, you're very good. Yes, Munna, I also know you're very good. All right, fine. So, Swami has a small doubt that is from a different chapter which I have recently completed. What is synaptonemal complex? Okay, so sorry for deviating. So, here I will explain synaptonemal complex. So, during meiosis, Swami, so during meiosis 1, that is especially in the prophase 1 stage, yes, so prophase 1 starts with your what stage? So it's leptotene, zygotene, pachytene, diplotene and diakinesis, right? So you have 5 substages, okay? So in case of zygotene, the chromosomes come together. So you have synapses. So the chromosomes come together, so that is synapsis. So when they come together, it is called as bivalent or tetrad, right? And in your pachytene stage, you have crossing over, right? So you have crossing over which leads to the formation of recombinant chromosomes. So when a chromosome crosses over, for this structure to be formed and to have a stability, you have a complex known as synaptonemal complex. The process of chromosomes coming together for crossing is synapsis. Okay, so when the crossing over happens, the stability is given by synaptonemal complex, because crossing over is not done, okay, let's cross over. No, you have a complex process of enzymes coming into picture. Stability has to be provided when the chromosome is exchanging its gene. Again, stability has to be provided. So that is done by your synaptonemal complex. Okay, Swami, yes? Okay, so these acids are present in your DNA as well as your RNA. So, Keshav, so if you look into this tabular column, the first one is for your RNA when they bind with ribose sugar. The next one is for your DNA when they bind with deoxyribose sugar. Okay, Keshav? Yes? Yes, clear? Okay, so done. I'm moving forward. So structure of a polynucleotide chain. Okay, so if I have 2 nucleotides, okay, so one nucleotide is with a nitrogenous base, a sugar and a phosphate. So how 2 nucleotides are linked together? So that is the question, that is how it forms a dinucleotide. So we are having a polynucleotide, right? So our DNA has 6.6 into 10 to the power 9 base pairs. So you have a lot of nucleotides, right? So base pair is a pair of nucleotide, right? So how are 2 nucleotides joined? Suppose I have adenine. Adenine is bound with my ribose, deoxyribose sugar. So this is bound with phosphate, okay? So this is for my conventional understanding I am writing. So I have 1 nucleotide, okay? So here I have another nucleotide. So we know A pairs with T, so your T is here. So thymine. So this is hydrogen bonds, right? So please don't get confused. So this is your H bond. So here thymine binds to my deoxyribose sugar plus my phosphate, right? So this is on a completely different strand. Our DNA is double stranded. On this strand I have adenine, this strand I have thymine. So both the strands are joined together by my hydrogen bonds. Suppose if I have guanine here, so guanine binds with deoxyribose plus phosphate. So how is this attached? These two has to be attached together, right? How are these two attached? So only if it attaches, then you have a huge polymer of your DNA, right? So these two are attached by your phosphodiester bonds. Okay, so you have 5' and 3' end, right? So between 2 nucleotides, it is joined by a phosphodiester bond. So here you have a 5' phosphate group. So here you have 3' OH group. So your nucleotides are added to the 3' end. So this 3' OH is pretty free and it is willing to accommodate another nucleotide. So that is how you have polynucleotides. Clear? Yes? So two nucleotides are linked through 3' to 5' phosphodiester linkage, because only in the 3' you have OH and it is willing to take in an incoming nucleotide. Okay, yes? Yes, Anthony Abhi, it's the same concept. Thus, more nucleotides are joined and you have a polynucleotide chain. So, polymer has a free phosphate moiety at the 5' end. So, your DNA has 5' end and it has a 3' end. Okay, so 5' end has a phosphate group, 3' end has a OH group. Okay, so here phosphate moiety, here OH comes from the sugar. So in between I have my nitrogenous base. Okay, so 5' end I have a free phosphate moiety and 3' end I have a OH group, and that is where my next nucleotide is getting joined. Okay, yes? Okay, so Shimpi, don't push Munna out. So I understand the problem of Shimpi, the pain of being the elder one at home. Yes, is that so? Yes, even I have younger siblings who are very torturous. So Shimpi, yes, I resonate with you. But okay, so being a teacher here, so Munna, okay, so you also listen to class and you should also take up all the Menti quiz. Alright, Munna, yes? Fine. So now Shimpi will also listen, Munna will also listen. Okay? Fine. So backbone of the polypeptide chain. So backbone is formed by phosphate and sugar, and by nitrogenous bases project from the outside. Okay. So nitrogenous bases are linked to the sugar moiety and it projects from the backbone. It's like this. So I have my sugar. So it is deoxyribose sugar. Then I have a nitrogenous base. It can be adenine, guanine or cytosine, and my phosphate group is here. So my 5' is owned by my phosphate and my 3' is the third carbon of my sugar. So first carbon, second carbon, third, 4, 5. So this is 5 carbon sugar. Alright, yes? Okay. Okay, Munna, fine. So yes, ma'am, I'm my younger brother too. Okay, yes. So here we go. So here let's pay attention to the structure. Ma'am, please repeat it. Okay. Yes, Shippi, I will repeat it. So this is how my DNA is architecture. So I have a sugar, I have a nitrogenous base. So sugar and nitrogenous base forms a nucleoside, right? Then they bind to the phosphate, that is nucleotide. So I am saying in a polynucleotide, I have many nucleotides. So this is one nucleotide, this is another, and this is another. So how are two nucleotides joined together? So here my two nucleotides are joined at the 3' carbon of the sugar. So this 3' has a OH. So this 5' has a phosphate group. So this 5' phosphate and the 3' OH joins together via my phosphodiester bond. Okay? Yes? So 5' has a phosphate group, 3' has a OH group, and this is my 5' end, this is my 3' end. Only in my 3' end, my nucleotides are willing to get added. Alright, Shimpi, did you understand now? Yes? Alright, Shimpi, okay. Cellulose is not polynucleotide, it is polysaccharide. Yes, so it has a lot of monosaccharides. Yes, Anthony, but good, you're trying to correlate. Okay, so here I have a pictorial representation. So let's look at the pictorial representation. Okay. So here it is my 5' end of the phosphate. So I have my deoxyribose sugar and I have my adenine, right? So if you look, this is one nucleotide. So sugar into nitrogenous base is nucleotide, and the nucleoside is attached to the phosphate, that is nucleotide. Right? So here at the 3' end I have phosphate and the 5' end I have phosphate and the 3' end I have OH. So 1, 2, 3, 4, 5. So deoxyribose is a 5 carbon sugar. Okay? So here I have 3' OH. So my next nucleotide comes and binds at this 3' OH. So between this 3' carbon of the sugar and between the 5' phosphate of the next nucleotide. So this is my next nucleotide. Yes, all of you are getting? Yes, and this too is your phosphodiester bond. So that is how two consecutive nucleotides are linked together in a polynucleotide. Clear? Yes, Vaishali, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. Okay, so I hope this is clear. So moving forward, so here we go. So in RNA, what is the difference? Okay. So, in case of your DNA, so we have 3' OH, right? The 2' carbon has only H. Okay. Ma'am, what is the difference between DNA and RNA? It is the sugar. In case of DNA, the sugar is deoxyribose sugar. So, deoxyribose sugar is also a pentameric sugar. Okay. So, I have my first carbon, second carbon, third, 4 and 5. So here we are having a OH, right? So here we have H. This is in case of our DNA. So now in case of RNA, what is the difference? So that is ribose sugar. So this ribose sugar is also pentameric. But here I have a OH, here also I have a OH. So in RNA, my 2' carbon also has a OH group. So whenever there is OH group, it is willing to react. Okay? Yes? Double-stranded RNA looks like DNA? No, not necessarily, because it has looks like, because from my normal eye, I cannot see a DNA and RNA, right? So I can just sequence it and see, only then I will know, right? And a double-stranded RNA has a different stability and a double-stranded DNA has a different stability. Okay, so see here in case of DNA, the sugar in the second carbon has H, so it is not reactive. So in a normal polynucleotide, the next nucleotide is willing to add at the 3' OH, because OH is a very vulnerable group. So whichever one wants to react, it will give way. Okay. Here, since 2' carbon also has OH, so this makes the RNA more reactive to reactions. This makes the RNA more reactive, thus your RNA cannot remain stable for a longer period of time. So this is the reason. So this is the prime difference between your deoxyribose sugar and your ribose sugar. Yes, clear? Okay, so moving further. So RNA, every nucleotide residue has an additional OH group present at the 2' carbon. So if you look, so this is my RNA, okay? So this is my ribose sugar. So first carbon, second carbon, third carbon, fourth carbon, fifth carbon is here. So the second carbon has OH, so this is more reactive. Whereas if you look at my DNA, so this is deoxyribose sugar. So first carbon, second carbon, third carbon, fourth carbon, fifth carbon, this has only OH at the 3' position. So this makes DNA more stable compared to my RNA. Also the presence of thymine enhances the stability to DNA compared to my RNA. Alright, clear? Fine. So here we move forward. So DNA is an acidic substance. It is in the nucleus, right? Of course, DNA is a genetic material. It is located inside the nucleus. So DNA is also acidic, okay? And it was first identified by a scientist known as Friedrich Michener. So when Michener identified DNA to be inside the nucleus and in the acidic condition, okay? So it is known as nuclein. So he named it as nuclein. So Friedrich Michener identified DNA present inside the nucleus, and he also found out that this DNA is acidic. Okay, maybe it stains acidic or it takes up acidic dyes, and he called them as nuclein. So Ravi Vatikan, yes, hello, thank you so much for your compliment. So please do like, share and subscribe, and thank you abundantly for joining my lecture today. Okay, yes. So, but one big disadvantage or the drawback was that we could not identify or isolate your DNA outside. So, thus elucidation took a lot of time. So, we know DNA is present inside the nucleus and it is acidic, it is called as nuclein. But we were not able to extract the DNA out of the nucleus, and thus the study of DNA took a bit longer. Okay, so the elucidation was a bit delayed. So, who were the two phenomenal persons who played an important role? So, it was James Watson and Francis Crick. So, these are the two scientists, and you would have heard their name a number of times, right? So, James Watson and Francis Crick. So, they found out. So, like if you give me one clue in a Mendelian genetics problem, then it is easy for you, right? So, you say, ma'am, you give us one clue, we will solve. So, similarly, actually James Watson and Francis Crick, they were actually working on DNA. So, I don't deny, they were also good scientists. They had given us a lot of conclusion. They were working on the DNA, but they couldn't arrive at a good conclusion. But on the other hand, in a nearby lab, you had Maurice Wilkin and Rosalind Franklin. So, these two people were also working on DNA. So, once these four people met together, so that time just like talking, right? So, if four friends talk and one friend shows you the notes, then you were like, oh my god, even I should do something like this, right? So, then Maurice Wilkins and Rosalind Franklin, they showed their X-ray diffraction data to your Francis and James. Okay? So, that is the moment they found out that DNA should be double helix, and these people got the Nobel Prize, but your Maurice Wilkins and Rosalind Franklin never got honored for their work. But it was from their clue that James Watson and Francis Crick found out that your DNA is double helical structure. Okay? Yes? But I seen a DNA as a colloidal white-like particle isolated from fruits. Yes. So, if I isolate DNA, it will be in a colloidal form. It will be in a fluid form that is very, very small. If you want to see DNA as DNA, then you need very, very high resolution microscope. Alright? Yes. Ma'am, is right lungs heavier than the other one? Yes, that is because of once again a balancing issue. So that I'll tell you, because tomorrow we are starting human physiology. So remind me that time that I'll explain why the right lung is much heavier than the left lung. Okay? Yes? Fine? Okay. Fine. So this is the story about James Watson and Francis Crick. So, hallmarks the base pairing between two strands of the polynucleotide chain. Of course, your James Watson and Francis Crick told that the DNA is a double helical model. What is one of the hallmarks? So, what is one of the best thing of your DNA that we have found out? So, we never created the DNA. So, we are just studying the creation, right? So, in biology, we do not create anything. We just study the creation, right? Fine. So, what was the hallmark of the DNA that they have found out? The hallmark is the base pairing between the two strands of the polynucleotide chain. So, DNA is double helix. So, here we have come to a conclusion. Sorry, my DNA is really worse. So, DNA is double helix, okay. So, these two strands are connected. That is why they are entwined. So, if I entwine my two fingers, imagine in between it is connected, okay. So, they have connected through hydrogen bonds. How? That is complementary base pairing. So, if I have adenine, it base pairs with thymine. If I have guanine, it base pairs with cytosine. So, these two are connected by phosphodiester bond, because this is one strand, this is another strand, but these two strands are connected via hydrogen bonds. So, that was the biggest hallmark of your double helical DNA. So, base pairing between two strands of polynucleotide chain, okay. So, also based on the observation, Okay, so how did we find out this hallmark? How did I know A pairs with T, G pairs with C, and it is complementary base pairing? Based on the observation from Erwin Chargaff. So he was also very important scientist, okay? Yes, but we created new things as biologists. Okay, actually we are exploiting the natural resource because we never created plasmid. There was something called as natural plasmids from which we manipulated for our use, okay? Yes, so also this was also based on Erwin Chargaff's observation. So what was Erwin Chargaff's observation? So here we go. Yes, so here we'll talk about Erwin Chargaff's observation. So this is a very important observation. So his observation is valid only for double-stranded DNA. It is not valid for single-stranded DNA or for RNA. Okay. So he said the amount of adenine equals the amount of thymine by the amount of guanine equals the amount of cytosine, and everything put together is a constant. That is 1. The amount of adenine equals thymine, the amount of guanine equals cytosine, and this equals a constant. Okay. Suppose if I say I have 60% thymine in my DNA, so then I can easily find out what is my adenine molecules because T pairs with A, right? Yes. So 60% is my adenine as well. But okay, so this I have found out, this is 60 and 60, but it is equal to 1, right? So if it's 60% is thymine plus 60% is adenine here, so this is 120, right? So here 40 can be my guanine plus 40 can be my cytosine, which is 80, which makes it whole to 200. It can be 100 also, okay, it depends. So, but 60 and 60, since I gave the example, it cannot be 100. So, here it is 200. So, this is how you calculate. This is Erwin Chargaff's rule. So, if at all based on this principle, they ask you a question. They say if you have totally 120 molecules of adenine, 120 molecules of purines, so then you should calculate. So, they will not say like this. They will only say you have 40 molecules of cytosine, then they will ask you how many adenine molecules you have. So from this you should be able to calculate. If C is 40, then G will also be 40. So 40 is 80. Suppose the constant is 1 here, because here it can sum up to 1. So adenine will be 10. So thymine will be 10. So together it becomes 100 because this is a constant, right? So here adenine percentage will be 10. Clear? Yes? Ma'am, is PBr plasmid modified? Yes, PBr is a modified plasmid. Okay? Yes. So is this clear? What is Erwin Chargaff's rule? Alright, fine. So, this is what is written here. So, double-stranded ratios between adenine and thymine, guanine and cytosine is a constant, and that equals 1. So, structure of polynucleotide chain. So, the base pairing confers unique property to the polynucleotide chain. They are complementary to each other. So, each strand from one DNA acts as a template for the synthesis of new strand. Okay. So, now I have 2 strands of DNA, right? Yes, my DNA is double helical. Yes. So, for my convenience, I draw it like this. So, I draw 5 prime to 3 prime, 3 prime to 5 prime. So, hold on, let me tell you what is this 3 prime to 5 prime and 5 prime to 3 prime. So, when this has to replicate, so I have my 5 prime to 3 prime, I have my 3 prime to 5 prime. Okay, so these 2 strands separate, then I have a new strand synthesis. So, each of these strands acts as a template for the synthesis of new strand. For this red strand to be synthesized, this is acting as a template because it is complementary, right? So, if here I have ATGC, so here I will have TACG, right? So, this is acting as a template. So in this case, so that is what is explained in this line. Okay, yes. Okay, so nucleotide has 3 components. Yes, so all this we have discussed. So only one more thing I have to talk about. The first thing about the hallmarks of DNA is complementary base pairing. Yes, so we know what is complementary base pairing now. The next one is DNA has antipolarity. So it is called as anti-parallel polarity. So I left this word. So we have double helical DNA, right? So this is 5' to 3' and 3' to 5'. So the poles are opposite, right? So this is meaning this has 5' phosphate, this has 3' OH. But here we have 3' OH and 5' phosphate. So this is known as anti-parallel polarity. Clear? Yes. So the total length, so if I have 2 bases, the distance between these 2 bases is 0.34 nanometer. Okay, so in a particular turn of DNA, so this is 1 turn. So here I have 10 base pairs and the height of this particular turn is 3.4 Armstrong. Okay, so all these numbers you should remember. So the total length of the DNA is 6.6 into 10 to the power 9 base pair is the total number of nucleotides. Thus the length is 2.2 meters. So this is the distance between 2 strands or 2 nucleotides. So totally we have so many nucleotides. So this into this gives you 2.2 meter. So 1 nanometer is equal to 10 to the power minus 9 meter. 1 Armstrong is 1 into 10 to the power minus 10 meter. Okay, so please remember all this. Clear? Yes? Know that? Fine. So salient features of double helical structure of DNA. So it is made up of 2 polynucleotide chain. That is why it is a double helix. The backbone is conjugated by sugar and phosphate. So nitrogenous bases project from the 3rd side. So it has 2 chains with anti-parallel polarity. One from 5' to 3', the other from 3' to 5'. The bases in 2 strands are paired by H bonds. So between A and T you have 2 double bonds. Between G and C you have a triple bond. Right? So the 2 chains are coiled in a right-handed helix. So meaning the helix is in the right-handed fashion. So this is my right hand. So if I draw like this, this is right-handed helix. Okay. So the pitch, that is height, is 3.4 nanometer. That is 10 into 10 to the power minus 9 meter. So 10 base pairs in each turn and the distance between 2 nucleotides is 0.34 nanometer. So what confers stability to the helix is the hydrogen bonds. They are stacked one over the other. So if I have one pair here, the other pair is here. It is stacked one on top of the other. The plane of one base pair stacks over the other in the double helix confers additional stability to the helical structure. Alright, so with this we are done talking about the polynucleotide structure. So in the next lecture I will talk about the search for genetic material. So we have a series of experiments. I will explain the experiments to you. Then we will talk about the most important, the process of replication. So please come to the next lecture because this is also basic. For the next lecture we want to talk about the experiments. So please get the experiments sorted and how the process of central dogma happens, that is replication, transcription and translation. So understand whichever background you can be from, but without these process your cell will not function. So please be glad to join my next lecture of my molecular basis of inheritance on Wednesday at 4 p.m. Alright, so please don't forget to join my 6 p.m. lecture. So we are going to discuss important questions, and tomorrow at 4 p.m. we are starting human physiology. For all those who are here, please don't get confused. Monday, Wednesday, Friday, 4 p.m. I teach a class 12 syllabus. Okay, Tuesday, Thursday, Saturday at 4 p.m. I teach a class 11 topic. So tomorrow being Tuesday at 4 p.m. we are starting human physiology. So please join me for the same. Okay. No, chemical coordination, I have taken 4 lectures. Alright, Dharani? Yes, if you're searching for my lecture, so find 4 to 5 lectures I should have taken. Alright, yes, so thank you so much for making the chapter very interesting. So wishing you more and more. So this is me signing off. So waiting to look forward to you at 6 p.m. So please join me for the same. Thank you.